Good afternoon, everyone. I'm Tai Cheng. Today, I'm going to talk about the most serious topic on modern oncology. How does cancer cells evade the immune surveillance of T and NK cells? So we'll go through the mechanisms and tumor microenvironments. And last, we'll talk about their therapeutic strategies. So first, why is this topic important? So even though chemotherapies and surgery are advanced day after day, cancer still remains a global crisis. So only in 1 year, in 2022, it took over 10 million lives. And in this case, immunotherapy serve as a beacon of hope, but there is a catch. The cancer isn't a passive target. They are evolutionary masters that change constantly. So the main objective of this research is basically these three. So first, we'll analyze the specific mechanism that cancer used to escape. And second, we'll talk about how does the cancer shield itself with the tumor microenvironment. And last, we'll find a way to fight back. So first, before we understand our prey, we have to understand our hunters. So our immune system basically relies on a double-layer policy. So first is the CD8 positive T cells, which belongs to the adaptive immune system. And they are basically the precise snipers, which identify the MHC class 1 molecules. And second are the NK cells. They belong to the native immune system, and they recognize missed self and stress signals, which they don't need prior exposure to initiate an attack. So by working together, they form a near-perfect surveillance net until the clever cancer finds a way out. So first, on the T cell side, the cancer uses mainly two strategies to evade the surveillance. So first is the invisible cloak, and second is the break. So some of the cancer will downregulate or even lose their MHC class 1 molecules, and in this case, they become invisible to the T cells. And if the T cells cannot see them, they can do nothing with it. And second, even being caught, some of the cancer cells will use like PD-L1, and by binding the PD-L1 onto the PD-1 receptor on T cells, the T cells are forced offline. And on the other hand, NK cells, they're harder to fool because they don't recognize what is they don't recognize what is wrong. They recognize what is missing. So, but the cancer are clever. They have two main strategies to deal with this. So the first one is decoy. So some cancer cells uses enzyme like ADAM10 and 17 to cut off the ligands. And when the ligands bind onto the NK cells, it makes the NK cells less aggressive. And the next one is some of the cancer cells will upregulate their HeLa-E, and when bind onto the NK cells, the NK send a "don't eat me" signal to the NK cells. And the next one is the tumor microenvironment. So the cancer doesn't fight on its own. It reshapes its environment. So the tumor microenvironment, short for TME, it's full of cytokines like TGF-beta and IL-10, which disrupt our immune system. And also, it's also low in oxygen and acid and toxic to our immune cells. And last, the tumor microenvironment also recruits some traitor cells, such as T regulations and MDSCs, which physically block our immune system from penetrating the cancer cells. And the last one is the most surprising, is that our immune system actually contributes to the evolution of cancer. So there's a thing called immunoediting, which means that our immune system kills the weaker cancer and then leaves the stronger one behind. So we've understood the mechanisms of the cancer. Now it's our time to fight back. So the first strategy, I call it releasing the brake. So by using some checkpoint inhibitors, such as anti-PD-1, PD-L1 antibodies, we can cut the link between PD-1 and PD-L1. And by doing that, we can bring our T cells back online again. But there's a downside of this method, is that it's not that effective on cold tumors, which means that when the tumor doesn't have MHC class 1 molecules, because if the cancer doesn't have this kind of molecules, the T cells cannot recognize it. And the second strategy is restoring NK policy and remodeling the TME. So we can use some chemicals such as ADAM inhibitors to block the decoy from forming. And by doing that, we can make our NK cells more aggressive. And the next, we can use CAR NK cells, which means engineered NK cells. They are tougher against the tumor microenvironments than traditional NK cells. So, in the future, the curing of cancer is less likely to be a silver bullet, but more likely to be a cocktail. We have to mix all kinds of therapies together to cure a specific cancers. And in conclusion, the escape of cancer is an evolutionary masterpiece. It constantly changes, it changes all the times, and there are lots of complicated mechanisms behind it. But if humans can figure it out, how does it work and what chemicals does it use, we will one day have a way to fight back. So in the future, The curing of cancer is less likely to be a one-size-fits-all, but more likely to be fully personalized. Thank you all for listening, and I'm now open for any questions.
Okay, thank you for your presentation. Your article is reviewing article. Okay,
Yes.
so how many literature you collected and reviewed for writing this article?
On the paper in that book, I go through around like 20, 21 at the S, and some of it's for the pictures. Some of it is for the pictures like this one and this one. So mainly I go through like Nature or Science, those kind of articles.
Okay. Could you explain how to analyze the specific escape of cancer cells against NK cells and T cells?
The specific what?
How to analyze the specific escape of cancer
Oh, the specific escape. Okay, so, all right, so let's go back to the T cell side. So just like we have mentioned, some of the T cells mainly recognize the MHC class 1 molecules, and some of the cancer will change the genes so they don't present MHC class 1 molecules on their cells, so the T cells cannot recognize it. So that is basically for the T cell side. And just like we've mentioned, some of the cancers will use, those are called checkpoints, checkpoint protein. So if that protein is attached on the PD-1, the T cells will be forced go into apoptosis. And as for the NK cell side, so for example, the first one, the ligand shading, so some of the cancer cells uses ligands like enzymes like ADAM10 and 17 to shade off ligands. And those kinds of ligands will attach on the NKG2D. And when being attached, the NK cells will be less likely to attack the cancer.
Okay. Could you explain how to explain the content of tumor microenvironment?
The tumor microenvironment, okay. So basically, the tumor microenvironment is when the tumor reshape its surroundings. So some of the tumor will make cytokines like TGF-beta and IL-10, which it is used in our body, but it's mostly used to stop the attack of our immune system. So when a infection is over, our body usually uses these two kinds of cytokines to stop the immune system, because if we don't stop it, it will attack our normal cells. But the cancer will use these kind of cytokines to stop the immune system from attacking the cancer cells.
Okay. Which immunotherapy is the best to detect, target, and destroy cancer cells?
So right now, just like we have mentioned, that in the future, the curing of cancer won't be a single therapy. So in fact, we have to combine all kinds of therapies together to get the most effective for curing one kind of cancer. But in current researches, I think the most effective one is the CAR-NK cell one.
And practically, how to choose which treatment method?
So it's basically based on what kind of cancer it is. So for example, if it is not cold tumor, like I don't know if we call it hot tumor or something, but if the tumor has not lost their MHC class 1, we can use the checkpoint inhibitors just by using the T cells. But there's a lot of complicated mechanism behind this. And there's not only these two strategies.
好，我们谢谢。